Hello everyone, I am Giulia Sassi, a PhD student at the University of Parma in Italy. I'm here to present a pipeline for the coevolutionary analysis of eukaryotic genes. Before going into the pipeline and its rationale, I will give a brief introduction to phylogenetic profiling, the comparative approach we use for coevolutionary gene analysis. A phylogenetic profile is a vector that describes the presence or absence of the same protein coding gene in a list of species. Phylogenetic profiling methods are able to predict functional interactions between genes that encode proteins from the same multiprotein complex or biological pathway by comparing their phylogenetic distributions. The underlying idea is that when two genes are functionally related, they should tend to be co-inherited, since the loss of either one of these genes would be damaging to that particular function. Since their very first representation in 1999, many variations have been proposed, both in the method of constructing profiles and in the metrics against which profiles are compared, to determine whether or not they are coevolving. In our recent paper, we describe a new large-scale method to assess similarity between profiles through the analysis of coevolutionary transitions, called cotransition analysis. We will consider cotransition analysis later as a final step in our pipeline. To perform a phylogenetic profiling analysis, three main steps are required. First, the definition of the species set under study. Second, the identification of orthologous genes in the species set. And third, the evaluation of a phylogenetic profile similarity. Let us consider the orthology in first step. The various methods of orthologous inference pay much attention to distinguish orthologous from paralogous genes. This is a fundamental point, given that orthologous genes originated by a speciation event and are considered to be the same gene in different species, so are likely to maintain the same function. Whereas paralogous genes are the result of an ancestral duplication event and very often differ in function. Anyway, we believe there is another crucial point that deserves further consideration. The most commonly used methods to retrieve homologous sequences in this type of analysis involve all versus all pairwise comparison using local alignment algorithms such as BLAST and Smith-Waterman. We have focused on how well these algorithms are able to retrieve distant homologous sequences. In other words, how sensitive are they, and thus able to return the most complete phylogenetic profile possible? To try to answer these questions, let us compare two types of analysis. This image shows the result of a BLAST diamond homology search with a human query gene in more than 1900 eukaryotic species. Significant sequences, shown in black, are found in only one group of metazoans. In the image below, the same search was conducted using Markovian models. As you can see from the extension of the green bar, homologous genes are recovered not only in the most metazoans, but also in some fungi and protist groups. Note that recovering more orthologous genes means have performed a more sensitive search. In this way, more complete phylogenetic profiles can be built, from which more coevolutionary information can be inferred. Based on these considerations, we designed a pipeline that develops all three steps of a coevolutionary profiling analysis, using hidden Markov models in the orthology inference step. We called it the Bro-Hammer pipeline. Bro stands for broccoli, the program we used to build the orthogroups. To perform our analysis, we selected a representative set of eukaryotic species from the comparative set in the EuProt database. To do that, we trim the eukaryotic set to the first NCBI taxonomic level and retain the species with the best Busco completeness score. We end up with 59 phylogenetically broad species. With the 59 proteomes, we run the orthology inference step using Broccoli software. Broccoli performs a diamond similarity search and then a phylogenetic analysis of the homologous proteins. Relying on the tree, builds a network of homologous relationships from which a machine learning algorithm identifies the orthogroups. We then use a hidden Markov model approach with the aim of extend the original species dataset and recover more distant orthologous sequences. First of all, we obtain a multiple sequence alignment of each orthogroup using cluster omega. Subsequently, we built the Markov model profile of each multiple sequence alignment with Hammer build. Every model was used as input for Hammer search to detect homologs across 1929 eukaryotic proteomes in OrthoDB, FASTA proteomes. As the final step, we applied cotransition analysis to assess the similarity between phylogenetic profiles constructed from the presence-absence orthogroup vectors in the 1929 species. To illustrate cotransition, shortened Cotra analysis, let's see two evolutionary scenarios, case 1 and case 2, with 16 species and 2 orthogroups. In black squares, the gene is present in that species. In white, the gene is absent. These scenarios have the same correspondence of 15 matches and 1 mismatch in the penultimate species. However, in case 2, there are 3 common events of loss of the ancestral gene, whereas in case 1, there is only 1 common event. As you can see from the table, the coevolutionary associations between gene 1 and gene 2 receive the same scores and significance by 2 standard similarity measures, even though the number of correlated events is higher in case 2. So, these global methods don't take into account the different coevolutionary information content between the 2 scenarios. Let's see the cotransition metric now. Brackets represent the transitions from presence-absence or absence-presence of the 2 genes during evolution. Cotr score considers the state transitions along phylogenetic profile vectors and measures the number of shared transitions among orthogroups. No coevolutionary information content is lost. In fact, Cotr score is different for case 1 and 2, particularly is most significant in the latter. So, with Cotr analysis, we are able to distinguish between a generical shared phylogenetic inheritance, as in case 1, and probable gene coevolution, as in case 2. To test the effect of orthology inference step on phylogenetic profiling, we compare the cotransition analysis from OrthoDB and Bro-Hammer orthogroups in a case study we already knew about. It's a multi-protein membrane complex widely distributed in eukaryotes. It consists of many interacting subunits, 12 of which are known and structurally characterized. By performing cotranalysis on the OrthoDB orthogroups, we were able to recover as significant transitions 9 out of 10 subunits, as shown in the network. With the Bro-Hammer pipeline, we recovered not only the 9 subunits found with OrthoDB, but also the other 3 that were missing with OrthoDB set. So, cotranalysis, preceded by Bro-Hammer pipeline, was able to recover all 12 subunits as significantly coevolving. We can better understand this result by taking a look at 2 specific subunits of the complex. Let's first consider subunit 6, which is correctly predicted with both OrthoDB and Hammer. By comparing the phylogenetic profiles recovered with the 2 methods, we see that the homologous sequences obtained cover the same evolutionary extension. The two methods give comparable phylogenetic profiles. Let's look at the results for subunit 10, correctly predicted by Brohammer but lost with OrthoDB. In this case, the use of Markovian models guarantees a gain in both the number and evolutionary extension of the recovered homologous sequences. The phylogenetic profile of this protein, obtained with Brohammer, is more complete and able to provide greater information content in terms of gene coevolution analysis. In addition to the specific case study, we assess the general performance of the Brohammer pipeline by comparing the cotransition analysis results obtained with those of the OMA and OrthoDB databases. We constructed receiver operating characteristic curves and measured the area under the curve parameter in protein pairs known to physically interact or not to interact in humans. We observed a different performance when Cotra analysis was applied to the three different methods. And in particular, there is an improvement in performance with Brohammer compared to both OMA and OrthoDB. In conclusion, we can say that, considering homology search sensitivity, pairwise alignment algorithms are not always able to return homologous sequences across wide species dataset. This is a key aspect in orthology inference step, and in our opinion deserves further investigation. The Brohammer pipeline is otherwise sensitive and able to return more orthologous sequences in our case study. And this seems to give an advantage over coevolutionary analysis of the corresponding phylogenetic profiles. Thank you very much for your attention.